Good afternoon, Korea. I hope you can hear me well. Good. So, I'm joining you live from Basel in Switzerland, early morning, not even 8 o'clock, but it's a great pleasure to be with you and to give you an update on ICH S10 on photosafety evaluation of pharmaceuticals since implementation of this guideline. So, this talk has actually 4 sections. Two at the beginning to wrap it up, what has happened in the past and how ICH is structured, or ICH S10, sorry, is structured, and then two main sections to focus on quantitative endpoints and how to use them doing photosafety evaluation and especially safety margins for potentially phototoxic drugs. So these are the two take-home messages after this presentation. So, a quick recap of past and present of photosafety regulations. Very briefly, and not going into detail, this is more for the documentation of this talk. What it's all about, it's sunlight, a driver of adverse skin reactions. And in general, without any drug around, sunlight alone can cause adverse reactions, and we call it often photosensitivity in the clinic. So, what is sunlight? Sunlight has certain ranges and components. Of course, the visible light, which helps us to see, but it also has an UV and ultraviolet part on the lower end of wavelength, the higher energy part, and these ultraviolet radiation is causing already adverse events. It causes sunburn, it causes photoaging, these two well-known phenomena. But ultimately, it causes not just damage, inflammation, but it also causes potentially skin cancer. And the eye is also a target organ of light. The ultraviolet part is not transmitting through the anterior part, but the visible light is reaching the retina. So the retina is also a very sensitive tissue and could be a target organ of sunlight-induced adverse reactions. So let's have a closer look. Also, exogenous molecules, which can absorb within the sunlight range, that's important. So if they can make use of a photon that comes in, can cause light-induced adverse skin reactions. So such acute reactions, similar to sunburn, are typically called phototoxicity. But we also have delayed reactions, typically involving the immune system, and this is then typically called photoallergy. And typically this also happens after a contact to an agent which is capable of transferring these reactions. So it's typically contact photoallergy. And those local skin reactions might be caused by contact to certain plants like giant hogweed, cosmetics, and very often they contained in the past bergamot oil or agrochemicals. And you see on the right hand side a typical picture how this looks like. The upper part of this leg was covered by the trousers, so no sunlight exposure, but at the feet there was exposure to a plant, and the light was just inducing this skin reaction very sharply where the light was able to reach the skin. So back to pharmaceuticals. Many active pharmaceuticals, the ingredients can also cause light-induced adverse skin reactions, limiting its clinical use significantly. And actually, both distribution ways are relevant. Systemic circulation to light-exposed tissues, so after oral ingestion, it's getting distributed to the skin, or local applications for dermal drugs. And just as a side note, accumulation or retention in skin can happen, e.g. via melanin binding, but is not essential for those phototoxicity reactions. It's just the blood flow through the capillary system in the skin which is typically sufficient to allow for these light-induced reactions. So you can look at accumulation or retention, but it's not a prerequisite to see acute phototoxicity. So as mentioned, there are also topical drugs, and their contact photoallergy as a delayed reaction is also important. But importantly, the areas of photogenotoxicity, photocarcinogenicity, and systemic photoallergy. So systemic photoallergy after taking oral drugs, for instance, those effects are of less relevance. Either they are rare, like systemic photoallergy, extremely rare, or there are no relevant test systems available which you could study these effects in. So these areas are covered actually by addressing phototoxicity in general and by avoiding those drugs or limiting their use, so to prevent also photocarcinogenicity on the long term. So ICH-10, how it evolved and what has happened since ICH-10 on the regulatory side. So before ICH-10 was initiated, so before 2009, there was already one OECD test guideline for the in vitro phototoxicity test, for the famous 3T3 neutral red uptake phototoxicity test. It was released in 2004 already. There were regional guidances, for instance, from the FDA and from Europe, from the EMA, and the kickoff for Photosafety actually was then on the ICH level. It was a revision of ICH M3 in 2009, where they added a section 14, Photosafety, a short paragraph only, but it emphasized that photosafety should be part of the non-clinical package, non-clinical safety study package, and it obviously required more detailed, harmonized guidance on how to address this field systematically. So from 2010 to 13, ICH 10 was developed by an ICH expert working group. I was representing FBIA in this expert working group. And since 2013, a number of things also then got lined up. There was a revision of the in vitro test, more for technical reasons, but it was also then harmonized with ICH 10, especially for the PIF value. There was another guideline on reactive oxygen species testing and another guideline for reconstructed human epidermis, so a 3D skin model and phototoxicity. This is a snapshot from the ICH S10 homepage, so at ich.org. And there is a section where also the status of implantation for Korea is mentioned on the right-hand side. So, very briefly, how is ICH S10 structured? What is the main principle? It is a stepwise photosafety testing strategy. This schematic is taken from the guideline itself. The upper part says initial assessment of phototoxic potential, and this is actually the chapter in ICH M3. If you have a UV risk spectrum, if you see the compound is absorbing within sunlight range, there is a certain threshold mentioned, then you should continue to experimental evaluation of phototoxicity. But if you see no absorption, you can go to the right-hand side and already conclude no further testing required. So typically you have to go to the lower part, experimental evaluation, and the guideline is relatively flexible here. It does not require certain assays per se, but it strongly recommends a stepwise testing from the simplest to the more advanced test. And the simplest one is, of course, in vitro testing, avoiding any animal testing and also not waiting for clinical testing. So typically it goes from the left to right, from in vitro testing through in vivo clinical testing to clinical testing. And as soon as one of these tests confirms a negative outcome, you're done actually. So if in vitro is negative, you can stop testing and conclude for the rest of your drug development it's not phototoxic. But if it's positive, it would be overwritten by a negative in vivo or finally a negative clinical trial. So why is this important? What agencies typically expect from companies today? So in reality, this has certain implications. ICH-3 in this short paragraph says the experimental evaluation should be finished before entering late-stage clinical trials, so exposing large numbers of clinical subjects. So by the end of phase 2, you need to be done with photosafety evaluation based on public data, or you have to accept protective measures until then. And during phase 3 and for marketing authorization, you need to be done, and if risk-benefit suggests that there is still a relevant photosafety risk, you have to put it into the label or the informed consent if you are still in phase 3. So this has strong implications. A label language or phase 3 with protective measures is something sponsors do want to avoid. It limits the use of the drug significantly and the commercial value. So you can't change at this time, end of phase 2, your drug substance. You cannot go back to a different candidate quickly. So the protective measures remain necessary. You have spent years, dollars, and won already, and marketing will be difficult. So selecting the right drug candidate upfront is absolutely key. So this is why companies do these things relatively early. So for instance, running an in vitro screening test to figure out if a drug substance is potentially phototoxic. So and this is a typical flowchart, how this could look like. It's very similar to the Novartis flowchart where I'm coming from, but many companies do something very similar. They start the spectrum very early on. If the spectrum is not showing limited absorption but showing relevant absorption, they would continue with an in vitro test. Some companies do this as a screening test, others wait for the first GLP batch to run this test. But if this test is negative, and negative means here below 5, based on ICH S10, the fifth value below 5, then you're typically done. And this is true for roughly 70% of the drug candidates during lead optimization, where you can still select your candidate. But if the fifth value is above 5, so formally positive, they would continue in case a molecule continues into the GLP phase and into first in human studies. They would consider testing it in vivo in an animal study. And the timing depends really on the company, whether they are confident on the negative outcome or they really want to know this early on. So it really depends on the special concept of the company and also the indication of the drug, how early this should be known and influence and decision making. But this is a typical way companies have actually adapted to ICH S10 and to try to figure out early on whether there is a potential risk and to mitigate and manage this risk throughout drug development to avoid an impact, especially a late stage impact on drug development here. So now to the two major sections of my presentation here, quantitative endpoints. And you've heard already two of them. One is the molar extinction coefficient and the other one is the pitch value, but there will be more details on this. So let's start with the molar extinction coefficient. It's also called molar absorptivity in physics and chemistry. And it's the normalized absorption property of a molecule. So what you measure is the UV vis spectrum in sunlight range, which is 290 to 700 nanometers. That's the definition actually. You could define sunlight slightly different, but this is the definition ICH S10 is using. So you see very often this 290 to 700 nanometers. And the highest value in this range, whether it's a peak or whether it's the lower end cutoff at 290 nanometers. So the highest value in this range will be used and we used to calculate the molar extinction coefficient, and this value should not be above 1000 liters per mole in centimeter. If it's not above 1000, you're done. Otherwise, you have to continue testing. And a side note, this measurement needs to be done under standardized conditions. It needs to be done in methanol. And and then it can be used um, as an experimental piece to decide which way of photosafety evaluation you have to continue it. So another quantitative endpoint, not just the MEC, the MEC value, but also the PIF, the PIF value. It's about the in vitro 3T3 nutrient uptake phototoxicity test. So I'm not going into details of this test. There is an OECD guideline for this. It has been um, described a lot in literature. But this is the main principle. It's a 3-day test in the lab. You're seeding these 3T3 fibroblasts. On the next day, you treat them with a compound on 2 plates. One plate is getting irradiated with a standardized simulated sunlight dose normalized to UVA. The other plate is kept in the dark, and then you just compare these 2 plates. And if you see an effect like on the right-hand side, that this neutral red uptake is limited because the cells are dead, the staining doesn't happen. You see in the upper part these colorless wells. So these cells died because of irradiation, so the compound was phototoxic, and you see how this looks like in a in a figure. So the right hand, the black curve, is the normal cytotoxicity profile of a compound. And if you see a shift to the left, this is additional toxicity caused by the irradiation, the simulated sunlight. And the ratio between the 2 IC50 values, so the factor, how much is shifted to the left, this is the photoirritation factor, the PIF value. And based on ICH 10, everything below 5 is considered negative. The OECD guideline also has a range of 2 to 5, which is equivocal phototoxic. But for normal outcomes, for regular outcomes in the pharmaceutical industry, a PIF value of 5 is the threshold. But if you see a positive, it also has a message. The higher the PIF value, the more positive it is, the more likely it is that this compound is a troublemaker, because it is a measure, a quantitative measure of in vitro phototoxicity. And so at Novartis, and we published this in 2014, we tend to have these categories weakly, moderately, and highly positive based on the given PIF values here. So now switching gears from in vitro to in vivo. In vivo tests are much less standardized. There is no OECD guideline for an in vivo fertility test, so it's basically all based on best practices and some publications. And I'm also quite optimistic that there will be a best practice paper, an industry-based joint best practice paper on in vivo fertility models, hopefully next year. But until then, this is what I can present to you here. Typically, you use rodents and you treat them for a couple of days and then you irradiate them. In some test models, you can irradiate every day because the animals are freely moving, not anesthetized, not restrained. You can just dose them, irradiate them, and then see what happens. Either clinical signs, which are erythema or edema, so swelling, but you can also do at necropsy at the end of this short animal study. You could do further investigation, especially eye histopathology. So this is a basic concept, using also simulated sunlight. And looking at the eyes, only if you have a compound which absorbs above 400 nanometers. So this is the cutoff for visible light. 400 nanometers and longer wavelengths are visible light, and if you have a compound which has not a flat absorption profile but shows some residual absorption there, you will also sample the eyes. So this is the photolocal lymph node assay. It's not a contact allergy test anymore. This is important. So some of you may think local lymph node means contact allergy. No. The photolocal lymph node assay for toxicity is a systemic test. You treat orally typically, but you use the endpoints from the local lymph node assay. So you see erythema and edema at the ear. You don't need to shave. You can just look at the ears. They have no furs. In these biopsy mice, you can treat every day. You can irradiate every day. You do this typically for 3 days, evaluate the animals clinically, and then have a necropsy. For the auricular lymph nodes, which are important secondary endpoints, but also for the ear weight, a punch out, and for the eye, if justified. So what you see here, and this is a very nice example of quantitative reactions, so a dose and time-dependent effect in this model. It's sparfloxacin on 3 dose levels. Sparfloxacin is our usual positive control, but here we have 3 dose levels for a validation study. And you see the erythema score from 0 to 3. And now you see a very faint green dotted line, which is emphasizing the low dose. Nothing happens through the 3 days here. But if you go through the mid dose of 100 mg per kg, you see it goes up on the first day after radiation. It stays at score 2. And on the third day, it's building up to erythema score 3. And if you go to 400 mg per kg, it already goes to the maximum erythema score on day 2, which shows nicely the dose dependency of the erythema score. But if you look at the ear weight, which is the upper right-hand box, you see it's a dose-dependent and light-dependent effect. So these are the irradiated groups, and you see nicely that only under radiation, the ear weight goes up, but also the lymph node weight goes up, and the lymph node cell count goes up. And you see this faint reaction here for the low dose. Actually, the ear weight went up at the low dose. So we didn't see erythema, but we see a significant slight increase in the ear weight, which nicely shows the dose dependency of this endpoint. So you would try to figure out where is your NOAL, your no observed adverse effect level for phototoxicity in such a study. So the low dose could be actually a NOAL, but looking at the ear weight, you would not call this NOAL, you would go one lower. So NOAL would be a clear dose level without erythema and edema in this model. This now is the long Evans-Red model, a very similar model, but in long Evans-Red, but it doesn't use auricular lymph nodes. It's a plain clinical science model. So you look at erythema and you look at edema, if you can see it actually visually. And this is the more commonly used model. It's established in a number of labs across the world, while the photologoliph assay is only available at selected sites in Europe. So this one is more generally seen. If you are an expert at an agency, you're most likely seeing more this model. But it's all about NOAL defining the dose, and especially for both models having TK ready. So knowing actually the Cmax for the NOAL, which is the most important parameter you would then use as translation to clinical exposure. So clinical exposure, this was the keyword. The last thing you could do on a quantitative level to follow up any phototoxic flag is a clinical study. Typically, you want to avoid this because you can only run this in parallel to phase 2 because you need to expose healthy volunteers or patients to clinically relevant doses. And it's quite late in drug development. So if you figure out your drug is phototoxic in this study, you're actually in big trouble because you wanted to avoid it, and in the last minute before phase 3, you learn it's remaining to be clinically phototoxic. It needs to go into the informed consent and into the label if you want to market it. So this is just an illustration. I'm not going into the details here. There are a number of literature references in here. So in the previous slides, there were references in here. You see also actually my name, Bauer in 2016. If you look this up in PubMed, you will find the manuscript related to the study type here, the clinical study. It is actually using the patient's skin as at baseline and after steady state and exposure to similar to sunlight. And if you see a change per patient in its sensitivity to sunlight, you conclude the drug has made the skin more sensitive to skin. So it's a different concept to the pitch value, but it's as quantitative as the pif value is. So it is the photosensitivity index, the PI here, which also is a quantitative endpoint and tells you whether a drug is phototoxic or not in healthy volunteers, and that's the figure here on the right-hand side. You see over time after exposure to a certain simulated sunlight fraction that there is over time an increase in sensitivity at 24 hours with the ciprofloxacin group, while the investigational drug and placebo stayed flat. So this is another quantitative endpoint based on the minimal erythema dose MED. So to wrap it up and to get closer to the conclusion, how to translate this into safety margins or exposure margins in 2 case studies. So looking at one drug which was confirmed to be clinically relevant phototoxic during drug development, it's vimorafenib. It's a published case. It's not from Roche. It's not from Novartis. Sorry, it's from Roche actually. It's on the market. It's Zelboraf, but it carries a label. The label says you need to avoid exposure to direct sunlight. The reason why it's on the market, it is a BRAF kinase inhibitor. So for a severe form of cancer, and there is a clinical benefit. But still the clinical use is limited because patients develop during phase 1 and 2 studies clinical signs of toxicity because the company believed at this time it is not phototoxic based on a negative in vivo study. So there was a disconnect and we followed up with this. This is a publication on the left-hand side. So we used the photo-LNA and a lot of investigations why this drug could be negative, actually false negative. And we figured out that it is positive in our test system. And in the end, we compared the light sources, and it was a light source in the initially used animal study, which was inadequate. This was done before ICH S10, notably. So based on ICH S10 using correct light sources, vimorafenib would have been clearly phototoxic in vitro, as we have seen it, but also in vivo, in both in rodent, in rats, and in mice. The other case study, which will show up on a figure on the next slide, is pradigastat. It is an investigational drug from Novartis for the cardiovascular area. Like vimorafenib before, it had a high PIF value. An animal test was triggered, and initially this test was negative up to 500 mg per kg, the top dose which was chosen initially. But looking at clinical development and the high doses used in the clinic, we decided to go higher in another study. And we figured out at 1,000 it's positive in animals. So the NOAL remained to be 500. This was the dose level in animals which was negative, and we had a related Cmax from this NOAL. And in the end, we were requested in those days to run a clinical photosensitivity study to really confirm this margin is sufficient. And the clinical study was negative. So the clinical study actually confirmed that it's negative in patients. And a 50-fold margin is sufficient here to argue that no label is needed. And you can look at these margins in a quite busy slide. So I'll take 2 minutes to go with you through the slide, and then we'll get to the end. So in the middle, you see the exposure in microgram per ml in different test systems. So in the mouse model, at the NOAL, so the no observed effect level, or the lower L, the lowest observed adverse effect level, which means phototoxicity. So go to the right-hand side, pradigastat, and you see actually, let's see if I have a pointer here. I have a pointer here. So you see the NOAL in blue. It gives 19 microgram per ml in these mice at the Cmax at a dose of 500. This was a dose where we didn't see any toxicity. The next dose at 1000 gives a dose proportional increase of 34 microgram per ml in mice and this was leading to erythema and edema. And clinically, in the clinical photosensitivity study, which was the steady state of the highest clinical dose anticipated, we only reached 1.3 microgram per ml at Cmax in these patients. So this clinical dose was negative in patients, and we have a 50-fold margin to the NOAL, which is the negative dose in animals, and we have an even higher margin to the LOAL, the phototoxic dose in the animals. On the left-hand side, with vemurafenib, which is phototoxic in patients, you see the NOAL in mice at a dose of 100 gives an exposure, a Cmax exposure of 55. The clinical dose was slightly above this NOAL in mice. It was 60, roughly 60. And the next dose in animals, at 350 mg per kg, was clearly phototoxic. So the clinical efficacious dose level actually is between the NOAL and the LOAL, and this was not enough margin. The margin is actually 1 for the NOAL. So this drug could actually cause clinically relevant toxicity. So this overview nicely demonstrates that safety margins are very helpful to look at and also can be used to justify that no clinical testing is necessary and no label language is necessary. And based on experiences, actually, a margin of 10-fold or above has often been accepted by regulators to be sufficient to not ask for label language or informed consent and protective measures. And this is also summarized in industry experience. There was a survey in 2018 organized by FBA and IQ2Safe, 27 companies, it's published. And there are a lot of conclusions, important also on how they run animal studies, as I just said, typically 3 dose levels up to MTD in either long Evans rats or biopsy mice, using simulated sunlight, normalized to a dose of 10 joules per centimeter squared of UVA, looking at erythema or edema in the eye, erythema or edema on the skin, and the eye when warranted due to absorption of visible light. This is best practices and common standard in the industry these days. And those companies who had the actually undesired opportunity to also follow up with in vivo toxicity, they also looked at margin of safety approaches. So some companies tried it and were successful with using the Cmax at NOAL versus the Cmax value in the clinic, as I just showed you on the figure. And margin of 10 to 15-fold were successfully used to justify no label language and no clinical risk, while in one case even a margin of 5 to 10-fold was successful. So the overall summary of this presentation. Phototoxic properties of systemically applied pharmaceuticals may be the cause of serious adverse drug reactions, despite being clinically manageable, which is correct, because for short-term use like antibiotics could be manageable and there are drugs on the market where this happens. For chronic indications, this is not feasible. So protective measures against sunlight can be applied, as I said, for short-term use. But both patients and health authorities are unlikely to accept it for chronic use. ICH S10 provides guidance on how to potentially, how a potential toxicity risk should be addressed. It's a tiered approach, so it's important it's a tiered approach, and that a next level test like in vivo is overruling the previous test, the in vitro test. So a positive in vitro outcome can be mitigated by a clearly negative in vivo test, or at least providing sufficient margins of exposure. Also important, toxicity is not like genotoxic or contact allergen. It's not a yes or no answer. It is not like present or absent. Toxicity is a dose-dependent reaction, and you have quantitative endpoints, and you can actually demonstrate that there is sufficient margin of exposure to manage such a hazard which does not translate into a risk. So safety margins based on Cmax at the NOAEL in animals versus clinical efficacious exposure at steady state in patients. This is the ultimate way to overcome such a flag and to still conclude that it's safe to go to patients and to have a safe drug to be marketed. And there is industry experience now more than 10 years after ICH-S10 implementation, indicating a successful adoption by sponsors and regulators globally. So here would end, just showing my acknowledgement slide with a lot of people over the last 15 years in this field from Novartis, but also from the guideline development and industry associations. And if you have any questions, I'm happy to take them now.